Okay, so today we're going to talk about topic 6.4, which is all about gas exchange. And as the picture implies, has everything to do with how we breathe, right? So gas exchange is all about the uptake of oxygen, right? So we need that for aerobic cellular respiration. And it's also about the release of carbon dioxide. And so that was a waste product, again, of aerobic respiration. And so if we look at the path, right, that air is going to take into our body, we know that we breathe in through our nose or our mouth. That air is going to go through the larynx and the pharynx and through a structure called a trachea. So the trachea is different than the esophagus. The trachea is just for air. And then it goes into these structures which we know of as lungs, right? So what might be new to us, right? So trachea, again, helps keep that airway open. It's made of a lot of cartilage. And then it branches out into something called the bronchi, which have further branches called the bronchioles. And then if we zoom in really far, there's a structure called the alveoli, which are these like balloon-like sacs. And so this is where most of what we're going to be working with is in the alveoli or the alveolus. So the alveoli, and so we're just kind of zooming into these structures here, are these balloon-shaped structures. And it's all about providing that extra surface area for the gas exchange. So that's why they're like balloon shaped like this. So the gas exchange is happening between the alveolus and the capillaries that are bringing in those red blood cells, right? So those red blood cells can carry oxygen and carbon dioxide. So the red blood cells are coming through the capillaries. They're going to pick up the oxygen that's coming in through the alveolus, drop off carbon dioxide that was a waste product from the rest of the tissues, and then go back along their way. So like most gases, oxygen and carbon dioxide are going to cross membranes via simple diffusion. They don't need any special protein channels. There's no exo or endocytosis or anything like that. So oxygen and carbon dioxide are going to diffuse directly across the membrane in and out via simple diffusion. And again, this is occurring in the alveolus. So when we look at the alveolus, there are two different cell types that you're going to need to know. So the first one is type 1. And so that is these guys here in blue. You notice that they have a long and elongated structure, which increases their surface area. Again, because that is where gas exchange is occurring, right? So these are the ones that are actually facilitating the gas exchange. And the other thing about the type 1 pneumocytes is that they are right next to the capillaries. So this is to decrease the distance that those gases are going to have to go into those capillaries, into those red blood cells. So the type 1 pneumocytes, again here, flattened, thin to maximize that surface area to allow diffusion to occur more rapidly into the capillaries. So that's the type 1. We also have these larger secretory cells, right? And so you can see that there's little vesicles coming off the Golgi and that something is being secreted into the interior of the alveolus. So it turns out that gases like to go through liquids better than a dry surface. So the oxygen and carbon dioxide dissolve more readily in a liquid surface than in a dry surface. So these type 2 pneumocytes, again, which are going to be these larger structures, they're secreting a fluid. And so sometimes you'll see the word surfactant, right? So they're secreting this surfactant that's going to coat the inside of the alveolus. And what that is doing is it's doing two things. It's going to allow the gas to dissolve and diffuse into the capillaries more easily. And it's also preventing these alveoli from sticking together. We know that when we inhale and exhale, we've got compression and expansion happening. So we don't want these alveolus to like get stuck when it's in a contracted position. So that surfactant, which is coming from the type 2 pneumocytes, helps to do that. So type 1, flattened structure, who's responsible for gas exchange to the capillaries. Type 2, secreting the surfactant, so they're going to be the larger secretory cells. So when we look at these under like micrographs, right, we can see that the type 1 pneumocytes have that long flattened structure. They're going to be along the edges versus the type 2 pneumocytes, which are going to have a larger structure. So again, those are the ones that are generating that secretory surfactant to allow the diffusion of those gases. Macrophages also populate the lungs because they're exposed to the air. So remember, the macrophages are the ones that are digesting any invading pathogens. So again, type 1 pneumocytes, these long thin cells that are facilitating the gas exchange. type 2 pneumocytes doing the secretion. So be sure that you can recognize those in micrographs. So again, type 1s here, long and thin, type 2, larger secretory cells. Okay, so let's talk about some terminology. So ventilation, which you know of as breathing, right? So we know that we breathe in, we inhale, and we breathe out. That's like exhaling. And so now we're going to use like some different terms. So inhaling is called inspiration, right? So inhale, inspiration in, exhaling, expiration, you're going out. And so what we're really going to focus on is the movements of the muscles that are around your lungs and rib cage that are facilitating this movement of inspiration and expiration. And so the important ones are the diaphragm and the intercostal muscles. And then we're also going to be looking at the movement of the rib cage itself. So before we get into that, let's just sort of take a lay of the land. So intercostals are muscles that are between the ribs. And they come in two flavors. External, which point towards the inside, and internal, which kind of points to the, like your sternum, right? So externals are going to point out, internals point in. It doesn't really matter, but you do have to know who is contracting when. So we have external and internal muscles that are in between these ribs. We also have the diaphragm, right? So the diaphragm is this large muscular structure that's sitting at the bottom of the lungs, and when it contracts, right, so this is its contracted state, is when you're breathing in. So when it contracts, it's connecting to the bottom of those lungs, it's pulling it down, those lungs down and open. So let's look at the different types of movement. When you inhale, your diaphragm is contracting. And when you inhale, it's the external intercostal muscles which are contracting. So I try to remember that these are basically inverses. That when it's inspiration, when I'm breathing in, it's the external that's contracting and also the diaphragm. What is also happening is that your ribs are pulling up and outward. And so you can kind of test this yourself, right? So put your hands on your ribs and take a really deep breath in. So you know, and when you do that, you're going to feel that your ribs are moving up and that they're moving out when you're inhaling, when you're doing inspiration. It's also intuitive, right, that when you're breathing in, the volume of your lung is increasing, which means that the pressure is going down. And that's because, just like the weather, air is going to flow from high pressure to low pressure. So when you breathe in and you increase that lung volume, it's going to allow the air pressure outside, which is higher, to populate the lower pressure lungs. So that's inspiration. So inspiration, your external intercostals contract, so does your diaphragm. Your ribs pull up and outward. Your lung volume is going to increase, which you can probably feel if you put your hands on your ribs when you breathe in. It's when you have a larger volume, it's going to lower your pressure, allowing the air to flow from outside into the lungs. So that's inspiration, also known as inhaling. Exhalation is the opposite, right? So exhalation is going to go with the internal intercostals. So when you exhale, it's the internal intercostals that contract. Both the diaphragm and the external intercostals are going to relax. And in this case, you know that your ribs are pulling down and in, and you can again put your hands on your rib cage, take a deep breath out, and you're going to feel your ribs pulling down. And so when you exhale, you can see that the volume over here of your lungs is decreased. And when I decrease the volume, I increase the pressure. And so in that case, their pressure is going to go out because now I have high pressure in the lungs versus outside. So air is going to flow out. Okay. So what you also need to know for the gas exchange is how to interpret some graphs and how to do some calculations. And two things that you will need to be able to do is to calculate a ventilation rate, which is just the number of breaths per minute, and something called a tidal volume, which is just the volume that is contained in a single breath. So if we look at this graph, right, you'll see graphs like this where you have some sort of lung volume in some sort of units. In this case, it's milliliters. I've also seen like decimeters cubed per liter. You'll have some sort of lung volume over here on the y-axis. On the x-axis, you're going to have time, usually in seconds, and then you'll have some sort of graph, right? So if we hooked up a person to a data logger, it would be recording their breaths, so how often they're happening, and also the volume of air contained in that breath. So when you're looking at these, one of these curves, like a full curve, is a breath. So in this case, when I'm going up, that's when the person is inhaling. When they're exhaling, it would be going down. So this is an inhale, exhale, inhale, exhale, inhale, exhale. So each one of these shapes would be a complete breath. So in this case, if I wanted to calculate, say, the ventilation rate, which would be number of breaths per second, I could look at the first part of this graph here, and this is what somebody looks like at rest, by the way. So if I wanted to count the ventilation rate at rest, I would go, okay, well, I have 1 breath here, 2 breaths here, 3, and 4 breaths. So this is 4 breaths in 2 seconds. And I know that if I take 60 seconds and I divide it by 2, that's 30 times. So 30 times 4 would be 120 breaths per minute, which is actually kind of fast. So that's how I would calculate that, right? So this is obviously somebody taking a big breath in. So, you could say if you see something like this is somebody taking a huge breath in, and this would be like one collective breath. So if you're asked like, what's going on in this graph, you could say, well, at 0 to 2 seconds, this person is at rest. For some reason, they took a huge breath. You might also see different graphs where the peaks are high and they're more frequent. So somebody's breathing really deeply, really fast. They're probably exercising, that kind of thing. So that's how you would calculate like ventilation rates and interpret what's happening here, right? So in this part, they're shallow breaths. There's not that many. So they're probably at rest here between 0 and 2 seconds, and they're also at rest between 5 and 7. Something is happening between 2 and 4 seconds. This is one big single breath. So if you saw a lot of these close together, they are probably exercising or something like that. So the other thing is tidal volume, right? And so tidal volume is the distance between the peaks. So for this breath, you would be looking at how much volume it is at the peak, which is around, I don't know, say 4.1, versus minus what the volume is in the middle, which looks to be around 3.5. So if I subtracted 4.1 minus 3.5, I might end up with something like 0.7125 liters. Something like that would be the tidal volume of this peak here. But that would be really different for this breath here, right? Because in this case, I have a tidal volume maximum, or I have a volume maximum of 6.5 minus 1.5. So this tidal volume would be more like 5, right? 5 liters or 5,000 milliliters total for that tidal volume. So that might be something that you're asked to calculate, say on like a paper 2, right? Okay. So to calculate the total volume, that's just a straight equation. You would take your ventilation rate, which in this case, like say we're doing the one at rest, it would be 120 times the tidal volume, which again, it's peak minus trough, which would be around 0.7125. So total volume would be both of those multiplied together. I have no idea what that number is, but that would be what the total volume is. Okay. So the last part of 6.4 is about the different kinds of diseases that you could find in the lung. So one of them is lung cancer. And so remember, cancer is dysregulation of the cell cycle, in which case you have uncontrolled growth of certain cells. So we already kind of know, due to public service, advocacy, that things like smoking or air pollution can cause lung cancer. And so if you have a situation where you're getting tumors throughout your lungs, it is going to cause difficulties with breathing. lung cancer does have a very high mortality rate. And so another disease of the lung that you might hear about is something called emphysema. And so emphysema is also caused by smoking, but what's happening is that the number of phagocytes, right, those macrophages that we talked about during the immune system, the ones that ingest uh, germs and create antigens. So those phagocytes, which are in the lungs, are going to increase in number because they sense damage. They think it's, a germ invader, and they're throwing huge amounts of elastase into the area. And so what happens is that the alveolar walls are going to be weakened and destroyed. And so now this person can't really breathe anymore. So this is what a lung with emphysema looks like. So this damage is irreversible because it's in the lungs. You're going to end up with shortness of breath. So it's a really bad disease to have, and that's why we, stay away from smoking. So that's going to be it for 6.4.